To understand AlphaFold 3, we have to look at the architectural constraint built into AlphaFold 2. That system was highly optimized for one specific task, predicting amino acid structures. It did this by treating proteins as rigid body frames, using strict geometric angles to calculate how a chain would fold. But cellular biology does not operate as a series of isolated proteins. Proteins operate within a dense interactome. They bind to DNA, process RNA, and are regulated by small molecule ligands and metal ions. This image illustrates AlphaFold 3's core breakthrough. Look at this predicted complex. We see a protein directly interacting with a double helix DNA strand, alongside bound small molecules. It achieves joint structure prediction across the entire biomolecular space within a single neural network. To achieve this broad capability, the developers had to completely strip away the rigid, protein-specific constraints of the previous version. Instead of hard-coding the geometry of amino acids, the new architecture requires the model to learn the rules of physical chemistry from scratch through massive-scale training. Predicting the physical structure of life required stepping away from a dedicated protein folding algorithm and building a universal molecular assembly engine. Feeding this diversity of chemistry into a single network introduces an immediate input problem. The system has to process drastically different structures, from long chains of DNA to tiny synthetic drug compounds, using an architecture that previously only understood strings of amino acids. AlphaFold 3 solves this with a unified tokenization system. For polymer chains, it compresses amino acids and nucleotides into single tokens, saving massive compute. But for non-polymers, like ligands and ions, they are tokenized atom by atom, providing maximum flexibility to model diverse chemical bonding. Before these tokens enter the main network, they are embedded with specific data. The system applies a hierarchical feature extraction, assigning user-specified covalent bond features and processing per-atom features, so the model understands the base state of the inputs. By unifying the entire chemical alphabet into a single language of computational tokens, the architecture can process the full spectrum of molecular inputs without relying on predefined, biased templates. In the previous generation, the architecture relied heavily on a module called the evoformer. Its primary job was to deduce 3D structure by analyzing deep evolutionary histories using multiple sequence alignments. That reliance created an architectural blind spot. Critical biological interactions, like an antibody binding to an antigen or a synthetic drug docking into a receptor, have no evolutionary history to analyze. This diagram illustrates the pairformer. It prioritizes direct geometric relationships over evolutionary history, removing heavy column-wise attention. Reducing sequence alignments to a simple 4-block embedding network, it then discards the alignment entirely, focusing computational power on a highly detailed 128-channel pairwise representation of tokens. The pairformer trades broad evolutionary context for deep, precise relational geometry. This allows the system to map arbitrary atoms that have never interacted in nature simply by understanding their spatial and chemical properties. Once the pairformer maps the relationships, the architecture executes its most significant shift. It completely replaces the previous rigid structure module with an unconstrained diffusion module. Look at line 1 of this diffusion module algorithm. Operating on raw 3D coordinates, it ignores biophysical rules, generating structure by iteratively denoising positions. The representations produced by the pairformer act as a continuous conditioning signal, providing the blueprint that guides the entire diffusion process. At the maximum noise level, the starting state of the structure generation is exactly this, an unorganized cloud of Gaussian noise. AlphaFold 3 treats molecular assembly as a generative denoising problem. It physically draws the atoms into their correct positions based on the exact geometric blueprint provided by the pairformer. This algorithm traces the sample diffusion rollout. In the high noise regime, where time step t is greater than 1, the model handles macroscopic architecture, like broad chain placement. Here, local physical impossibilities like atomic clashes persist, as the model hasn't committed to exact microscopic geometry. The critical biophysical phase transition occurs precisely when the noise schedule crosses t equals 1. Entering the fine refinement regime, where t is less than or equal to 1, the remaining coordinate perturbations are extremely small. This allows the network to rapidly correct local violations, enforcing strict physical chemistry and resolving atomic overlaps. This delayed commitment is deliberate. It ensures the network finds the optimal global architecture before it resolves the microscopic atomic fit, completely avoiding the local energy traps that stall traditional physics simulations. Because diffusion is an unconstrained generative process, it carries the risk of hallucinating physical structures. Verifying the output requires precise confidence calibration. To evaluate local confidence, the model calculates the predicted local distance difference test, or PLDDT. This metric measures the network's certainty in the predicted position of each individual atom. This predicted aligned error chart evaluates global packing by measuring relative distance error between tokens. Calculating this per token provides mathematical proof of structural reliability when evaluating how synthetic drugs and metal ions interact. Despite its structural accuracy, the architecture hits a strict biophysical wall, known as the variant predictability deficit. When researchers introduce a single-point mutation that is experimentally known to destroy a protein's stability in vivo, AlphaFold 3 fails to predict the collapse. Instead, it almost always outputs an identical, highly stable wild-type conformation. Plotting AlphaFold's confidence against actual experimental stability changes proves the model's confidence does not reflect physical folding defects. Because AlphaFold is a pattern recognition system trained on static structures, it cannot calculate these thermodynamic pathways. To bypass this static limitation, computational biologists take the high-confidence static coordinates generated by AlphaFold 3 and pipe them into traditional physics-based molecular dynamic simulations to observe how the molecules actually move. AlphaFold 3 has mastered the static blueprint of the entire cellular interactome, but uncovering the kinetic and thermodynamic reality of life still requires a synthesis of this new AI architecture and foundational physics.